right, guys. Can you hear me? Can you hear me okay?
Yes.
Okay, very good. So you can see the screen, right? Can you see the screen, share screen and everything?
Yes.
Yeah, okay, very good. So this is what I introduce myself. Typically, you know, when we meet together, have a conference, we start with, hey, my name is Nam-Joon Cho, and then we introduce ourselves. But nowadays we have a new era of COVID-19 with a new greeting. Can you hear me? Hello, and so on, because of this June conference. Today I'm gonna talk about the emerging problem that we have the problem right now, and then I'm gonna discuss with you about, you know, what is the solution. And then start with, I briefly introduce my research area, which our department in our university asked for me to prepare for the research area, research interests, so I can actually throw in using this topic, and then you can actually see my email and then my website. If you're interested, please contact me. So there is a two very different area of research we have. If you go to our website, that project divided by 12 different interlinked together, which is every topic is related with the lipid assembly. Pollen composed of a lipid nanoparticle right now vaccine material, that is the LNP, that's what we do. And also the virus particle, we are talking about lipid envelope, that is exactly same as a lipid nanoparticle. So today I'm gonna talk about the research highlight of this talk is basically, instead of we target small molecule like developing inhibitor, we directly target lipid envelope of virus. That's what we call lipid envelope antiviral disruption. This is a lead technology we develop. We make the word and coin the name. And then this is the first-in-class lead peptide platform that we can quickly design and identify product candidate with a broad antiviral and bacterial properties. And then we are hoping that right now current situation, which I'm going to explain about COVID, but in a future disease X threat, we're hoping that we can actually using this broad spectrum antiviral therapy. And then the multiple milestone, we are basically having funding not only from Singapore, from Korea, from US and NIAID in order to develop this very exciting program. So let's start with this whole mapping. And then we saw in Korea, for example, we had the MERS, but at the time it was a very, very short period of time we had the infection. In Singapore, yes, we have a dengue outbreak and so on and so forth. But when we remember, you know, 2 years ago, 1.5 years ago, 2 years ago, beginning of, you know, 2000, we never expect that, you know, this SARS COVID-19, you know, happening this way, right? Evolving this way. And then nowadays we are talking about live with the COVID. Really see the whole history. Let's take a look at the history from 1520 to 1800, 1918, 1957, 1968. But what do you see here? You see the number one, you see that the time gap was very broad. And then there is a pandemic, epidemic is coming on and off. But recently, after the HIV and dengue, it became very, very fast. And then those term is very short. And then what I'm seeing here is we try to find, instead of develop when those things happen, develop each case, response to the each virus, I try to find a way out of how can we actually treat this virus as a whole, and then find the common denominator to target that common denominator, which is, if you actually see it, they have a one common denominator, which is lipid envelope. I mean, that's why the right now the current design of a vaccine from Pfizer and Moderna, they're using the lipid nanoparticle, which is lipid envelope, lipid assembly. So if you actually see this whole mapping of a pandemic epidemic situation, about 85% plus emerging virus pandemic involved with the lipid envelope. And then disease X more likely will come again, and then that have to be responsive with the lipid envelope. That's why we are targeting lipid envelope. And then if you actually see that this left picture is showing the flavivirus with the blood-borne, which is Hep C, or mosquito-borne flavivirus, including dengue, chikungunya, yellow fever, JEV, and so on and so forth. But this is not everything. We are talking about dengue and so on. When Zika happened, this left figure is basically, they don't even mention about Zika, which is a very old virus. But if you mapping out, if you open the mapping, you never heard about Ehrlichia virus. You never heard about Potosi virus. And there is a lot of unknown disease acts virus can be happen. So you cannot just wait for the virus outbreak, then prepare for the solution, because you're not going to be winning this race. So, when this virus pandemic happened, I had the contact by the Nature, one of the editor and then the reporter contact our whole collaborator and our team, discuss about how we're gonna be prepared or proactively stop viral pandemic. And then one of my answer, if you actually read this is a recent article on April 15, I said, hey, we need to really prepare for the next pandemic, stocking the file for the drug. And then, what kind of drug we were talking about? I said, hey, we are developing vaccine, but there is a lot of different type of drug. For example, repurposed drug, prophylactic, or what I developed is the broad spectrum. One drug for not just one bug, one drug for multiple bugs. How this work? Because right now, as you see that, for example, for instance, Singapore, we got a vaccinated rate of higher than 80%, I believe. And then today or yesterday, we have the high number record of people got infected. As the symptom is very minor, 2,000 plus. If you convert the population wise, it's a 40,000 in Korean population. In Korea also, there is about 51% of the people who got vaccinated, but still we have lots of patients got infected. So there is plenty of opportunity this kind of virus evolve into the strain that currently vaccine cannot stop. More contagious coronavirus variants can be happen. So vaccine and antiviral drug is not enough, and then which is we are learning. And what is happening? What kind of class of drug people developing? And then something happened. This is a virus infection happened. People take a look at this virus life cycle. Number one, virus entry. Number two, reverse transcription. Number three, integration. Number four, transcription, translation, cleavage, assembly, budding, and so on. So you gotta produce more virus. What people do is one or two pathway, they try to block, inhibit. in the molecular level. So that's why if you mutate, then you need to do it all over again. That is including the fusion inhibitor, protease inhibitor, and so on and so forth. So why don't we basically treat this virus particle and then target and then just identify the common denominator, which is lipid envelope, target that. So that is what I've been working on for a long time. And then one of the mother peptide we're using is from Hep C. It's not from nowhere. It's from Hep C, which is one of the non-structured protein, NS5A. They contain very interesting, very special peptide, 27 amino acids of amphiphilic helical peptide, which that is a membrane association, known as a membrane association peptide. But not only that, this peptide is resembled to the antimicrobial peptide, but it's totally different behavior they are possessing. What they do is basically they can actually lysis the lipid envelope of a virus with a certain size. The meaning is they can actually sensing the curvature of lipid envelope. They sensing it, they make the pore formation, they lysis it. So they can actually active against a virus size below 175 nanometer. Above, they cannot do it. So that's why the safety-wise, the cell membrane, they don't have any efficacy. The meaning is very safe to the conventional cell, which is 10 micrometer plus. However, the small size lipid membrane, they have their mode of action. So we test the various virus, and then you can clearly see that in the artificial as well as the actual virus, it's working against below 175 nanometer. And the negative control, of course, if you have a bigger size, which is some of the bacteria, some of the viruses, it does not work. So the mechanism of this lead peptide, fighting infection by size, not by the pathogen, so that as long as they have a lipid envelope, if you actually lysis out, then you can actually lower down the viral number in your body. Then you can actually develop, based on this one, you can have a cure. So, I mean, we are engineer, I'm a material scientist as well as a chemical engineer, processing engineer. So what we do is we develop the model membrane system, resemble the virus, and we develop new way of assay to basically mimic artificial viral platform so that we can screen out our drug, and then we do the drug evaluation. So it was a very precisely we can done in a sense that we can actually simply make the synthetic lipid envelope particle platform, as well as we can inactivate. In this particular case, it's showing as dengue or COVID-19. Attached on the platform with the fluorescence label, you can clearly see that we can actually label with the cartane. You can incorporate the dye inside, and you can actually label the lipid envelope so that you can track this single dot as the either artificial lipid or virus, inactive virus. You can clearly see that once you apply the drug, you see that the intensity drop. From there, you can actually screen out the drug. With this platform, you can compare with the various of antimicrobial peptide as well as how those peptide is sensitive enough in biophysical fashion. This is very important and have a large implication because instead of directly working with a cell platform in vitro, by using this kind of antiviral biophysical platform, you can save tremendous of time and effort to screen your drug Then you using this platform, you can actually move that to the in vitro cell study, including dengue, yellow fever, as I mentioned, broad spectrum, chikungunya, JEV, Ebola, Marburg, and so on and so forth, based on your criteria, which we did. With that, we developed this lead peptide generation 3, third generation peptide, in order to countermeasure for COVID-19. And then you can clearly see that in here. We develop about 11 different kind of antiviral agent. And then we try that, we see that EC50 is low nanomolar range, which is 30 nanomolar or so, which is compared with any of the conventional antiviral drug. This is the most potent drug of all time, which is, I'm working on the IND right now, process in with NIAID. And then in here, the most important thing is basically PK/PD, biodistribution, as well as how much they can actually circulating. And then you can clearly see that this peptide, which is 25 milligram per kilogram, for the IV administration, you can actually stay in the body for 24 hours, not only within the body, blood, spleen, and liver, and then also brain. It was a very exciting point. This peptide can be actually penetrate in the BBB. Before that, we also finished the toxicity study, because the one of the very important thing in biology is toxicity as well as biodistribution. The meaning is the efficiency and toxicity. Above all, even though the mechanism is very interesting, the mode of action is very unique and novel, end of the day, if you want to use it for clinical trial, have to be very safe, number one. Number two, stay in the body for a long time, so have efficiency. So we using, as you see, that 30 nanomolar, that is the range of the concentration we use. However, we go up to 45 milligram per kilogram. It's very high dosage. We don't see any toxicity study within those candidates. And this is the basically, indistinguishable to the control for the hematological result, the various. And then also for the parameter we test, even at the high dosage, it's a similar coagulation parameter you can actually get compared with the control. So we did this one directly. We are comparing with the remdesivir you see on the TV, and then people try to use remdesivir for the repurposed drug. And then also Fizenon is another drug, 36 amino acid, which is used for the HIV before, and then they try to use it for the COVID-19 because we don't have a solution yet. I know a lot of company and big pharma is working on it, but this might be another solution, because we are not only finishing targeting Zika, and we are moving to the other virus, because the mode of action is broad spectrum. So in the model of Zika, which we are working on COVID-19, but it's very difficult to access with the big animal model, which we are doing it right now. And it takes long time, but you can actually explore it, and you can get the idea of if you're using other virus, which is exactly work with the mode of action is the same. So in this particular case, the study, the evidence for the empiric helical peptide we're using, and then we basically use this one, number one, for the biophysical design, and then we demonstrate viral envelope targeting. Number two, we demonstrate this one also penetrate or passing blood-brain barrier, and then working against in the blood and brain, they inhibit the viral spreading and then inhibit the viral number. Then in a therapeutic solution, we basically check the decreasing viral load, prevention of the brain damages, reduce of the clinical symptom, protect against research infection. So this is the 4-5 different way, and then we using it 2 different fashion. One is a prophylactic, other one is a treatment. So which pathway we're gonna go, we didn't decide yet. But as the one of the reason I'm showing this mapping, every single biologic study or virus study people do it start from, and then they start from here, therapeutic evaluation, clinical sign, viral load analysis, and so on and so forth. What we do, we start from biophysical design. That's the very different design of study, because we are engineer, we can actually do peptide engineering, lipogen rupturing kinetic, biophysical study, mode of action study, and then that everything, that including the fast track pathway and mechanism studies, so that we can efficiently design this antiviral strategy. Not only that, we can actually save tons of time when we actually do the in vivo and in vitro studies. So in this particular case, you can clearly see that our peptide, which is even 10 nanomolar to 50 nanomolar, you basically decreasing the cell death. So protect against the Zika-induced death in the neuronal culture cell. And then, and also we showing that the inhibitory effect against Zika, chikungunya, dengue, JEV, yellow fever, and so on and so forth. The index, therapeutic index, is up to 5,400. And then the very important thing is when you're using the mouse model study, we can do both. The meaning is prophylactic as well as therapy. Our animal model is basically designed that after the infection, 6-7 days, 100% die. However, in this particular case, typical biologic study design people do is basically they mix Zika virus with the drug, and then they start injection. That's the day 0. What we do is 2 different fashion, because you got infected, and then the virus or virus is proliferating. Their level of a viral particle is very high at day 3. So we start doing our injection day 3, day 5, day 7, and then we check day 4 and day 6, and so on and so forth. So in this particular case, we demonstrate that clearly survival rate, we can recap 86%. That is basically 18 out of 22 mice is survived. Or we can actually using a prophylactic. For example, unknown virus outbreak is happened, and then we just take the medicine, then go into the site prophylactic way. And then in this particular case, also we see that we can actually increasing the survival rate dramatically. With this study, we check the viral load and neuroinflammatory analysis, not only serum, spleen, optical nerve, also with the brain. It was very exciting. We never expect that, this kind of peptide can basically penetrate BBB. Not only that, passing through BBB, and then they basically efficiently actually lower down the virus, as you see that here. It's about 3 rows. The meaning is 99%, 99.99% of the virus you can actually kill it at day 7. So with that, we basically study with this peptide how much they can actually influx to the passing through BBB, comparing with the positive control of the dermorphin, which is known molecule to penetrate BBB. And then we basically study other thing because one of the reviewers saying, hey, when you infect by the Zika, the BBB is loose now, so that's why it can be, potentially penetrate into. That's why we test with the day 3, which is we know that BBB is intact, as you see that here, and then after day 7, they start to lose the BBB penetrate by the we studied the fluorescence study, so that we confirm that this penetration of BBB is not because of the injury. It is a protect against the BBB injury. So with this fundamental study, this lead peptide of interest, we design new one now. We test with Zika, 4 serotype of dengue, yellow fever, JV, and so on and so forth, including the different strain of COVID is working against very nicely. And then the below 3 is basically negative control, which is non-envelope virus, it doesn't work. Also non-RNA virus is not working, or the bigger size is not working. So there is a very clear mechanism of mode is action. We are defining that we're working on this very exciting project. So this peptide therapy not only inhibits the Zika infection, but for the most of the flavivirus, or even further for the lipid envelope. And then this also address the other Zika related medical complication. Then we can actually use this one for the tissue, for the pregnant and sexual transmission, and also eye infection. So the motivation in here is that this is a new class of antiviral strategy and a whole new mode of action, which is developed only in our lab. And then we are pushing it, this mechanism, and then working with the various of group, international organization to pushing this effort. And then the one of the reason I give you a presentation this one today, I believe I have about 1 or 2 minutes, because even though we are engineer, we put actually our whole effort together with the first line virologist in order to solve this problem. So this is my excitement, actually solving this whole level of global health problem, and then we're very exciting to contribute in a sense. So if you guys wanna join this effort, more than welcome. And then also the whole topic we are interested in, then you can actually see our website, which I showed one of the things is replace plastic. We are using natural material to replace plastic. I believe we demonstrate a lot, and then the paper going to be come out PNAS very soon. It just accepted a few days ago, talking about how we using this natural material to actually make the packing material, including straw and box and so on. So this is a very exciting moment. And then I finish my talk by now, and then I will take the question if you have any. Thank you very much.
Thank you very much for a wonderful presentation. So I'll open up for a Q&A session now. So please, if you have any question, feel free to speak up or use the chatting room to ask the question. Really, I have a question. How can we know that a pre-developed pre-screening or a therapeutic method will be effective in the next pandemic viruses?
I mean, that's an excellent question, because number one, we don't know. That's why we need to have a common denominator. I'm not saying that I can actually have the solution for all the virus, but at least I know most of the epidemic pandemic virus is the lipid envelope one. More than 90%, 85, 90%. So once this happens with the lipid envelope virus, which is more likely happen, and then one other link is, I didn't mention about it, one other link is a very exciting link. You see that right now all the people crazy about RMP. Everybody talking about mRNA, RMP, vaccine, blah, 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 all the drugs. Why is RMP? I mean, this is old technology, but lipid envelope of a virus, exactly, basically same as RMP composition. So if you link up together, you can actually see the how this vaccine working against virus is so efficient. This is related with the all interfacial science, vesicle fusion, is RMP fusion, and virus fusion is all related. This whole mechanism is very linked together. So a lot of biophysical people, they are basically study this whole mechanism of RMP fuse to the cell and translate an infection, which is the same pathway.